Welcome to Boom, where we have
biomechanics on our minds.
Boom.
Boom, boom, boom. Boom.
So today we are really excited to be talking with Professor Maria Holland. Maria is the Claire Booth Luciff Assistant Professor in Aerospace and Mechanical Engineering at the University of Notre Dame. And I'm really happy to have you on, as you were also a PhD student in Ellen Kuhl's lab while I did one of my first rotations here as a student at Stanford there. So it's really cool to see where you are now. And we are also really happy and honored to be included in your outreach initiative through your NSF Career Award, which we found out you were awarded. So that's really exciting. Congratulations.
Yeah, thank you. Yeah, it's very exciting to be sort of full circle, like you said, Melissa, full circle regarding sort of grad school, where we had met at sort of the beginning for you, and now maybe you're getting closer to the end. And then, I'd reached out about the career, and after an unsuccessful try, here we are, and it was awarded. So I really appreciate the opportunity to be on here and talk with you guys.
Yeah, thank you.
Yeah, it's so great to meet you. I'm sad that I missed you here at Stanford, but excited to be connecting here.
Yeah, and your voice is very familiar to me from the podcast.
Yeah, it's funny. We get that we recognize your voice, but not your face.
Yeah.
So nice to meet you face to face.
Yeah.
Okay, so as an avid listener of Boom, you can guess what our first question is going to be, and it's a question I love hearing. So, when did you first know you wanted to be a biomechanist?
Pretty late. After I joined my group in graduate school. So, so I was not. So I had sort of a long and kind of reluctant path, I guess, to biomechanics. So in high school, I really liked physics and calculus, but was really, I hated biology. I really hated sort of the messiness of it, both I think physically, getting my hands dirty, but also that biology is so complex. And I also hated what felt like the rote memorization of it versus sort of the first principles aspect of physics and math. So I started out as a physics major in college, and then and specifically was not interested in mechanical engineering because I didn't like cars, and that's what I thought mechanical engineering was.
Cars and trains.
Yeah. So, oh my gosh, the engineering
biology is
books that I
affecting
now have.
problems.
Yeah. My son has these books that talk about engineers, but they're all about trains, and I'm like, no, this is not. Was not interested in mechanical engineering, didn't like cars, but then I got involved in this student group that was doing sustainable energy projects in China. And I traveled with them a few times to work on these sustainable energy engineering projects, and I just sort of saw the potential of engineering, what you could do with it, and that was exciting to me. And it wasn't cars, so I changed my major towards the end of my sophomore year. And then when I was looking at graduate schools, I specifically discounted any lab that had bio, micro, or nano in the in a prefix of their research field. I was just nope. No, I want to do really I don't know, straight solid mechanics. I don't know, I like FEA. I finite element.
Yeah.
And just the bio stuff, I was like, nope, not that. For some reason, a lot of people do this, but it's not interesting to me. Not what I want to do. And then I got to Stanford, and I met, so Melissa mentioned, Ellen Kuhl was my PhD advisor, and I reached out to her because I was interested in finite element analysis, and that's what she did. But with bio applications, and I was pretty hesitant about that, but talked to her anyways. And she convinced me, which was very effective for the record. She convinced me that in her lab, I would sort of learn tools and skills that could be applied, in my PhD, I would apply them to biological questions. But after my PhD, I could apply them to anything. And I was like, that sounds good. I can sort of put up with it for a few years. Hindsight, man. And she got me started on this project sort of related to brain development, and I started working on that. And over time, I think it just really grew on me. So I'm still working in the area. I haven't decided to take those skills elsewhere and apply them elsewhere. And I think now I just have a little bit more mature perspective on biology. But like the messiness and the complexity is sort of exciting rather than frustrating. Like there are just so many questions and so many things to learn. And I also see sort of the ways in which, I mean, because I'm studying biomechanics, not pure biology. And so there are like first principles and some of those things that sort of go into how the body works. It's not what I learned in my sophomore year of high school in biology class, but there are sort of these universal principles and laws and things like that also govern biological systems. So it sort of won me over. And now I'm really enthusiastic about biomechanics, but it took me a while.
It's so fun to see that change in perspective from being resistant towards that messiness to now embracing it and being excited by it. I feel like I can definitely relate. I really wasn't interested in biology in high school and thought it was just memorization. But now it's so fun to be in the field and really, like you're saying, also applying these mechanical principles to the body in ways that can help people. And
and I think it's a good lesson of like going from what you know, like working from what you know and like pushing a little bit beyond, a little bit beyond, a little bit beyond. And like, then, like you don't have to jump right in and love it right away. Like you got to work from where your first principles were.
It's sort of little steps. And I think the other thing about like, from the outside of a field, things can look very different than from the inside. So from the outside, biology looked to me like, I don't know, here's a textbook full of like definitions and just memorize what an integrin is and like what a mitochondria is. And like, that's sort of what it felt like. Like these words are made up, like what are these different things? Like, it just felt like I didn't have any tools to grab. Like now as a professor, as an instructor, I think of it like I didn't have the scaffolding, right? I didn't have like a place to put this information. It was just like 1,000 Post-it notes, like all over a wall. And like, even without the sort of like crazy, like serial killer investigator, like string connecting thing, it was just Post-it notes. And so now I'm like, okay, so I can remember like what actin and myosin are because like I understand their function. And so like, it makes sense to me that okay, I had to sort of learn these two words, but like I understand sort of what they're doing and why they're doing that and that sort of stuff. So from inside, like with an expert sort of point of view, not that I'm the world's best expert on biology, but it becomes more like logical and doesn't seem as random as
it's so true. I do remember making flashcards for all these different terms, but now I'm like, all of these things are integrated together. And when you learn that and how they function together and what their purpose is, it just makes so much more sense. And it makes me want to learn it more rather than, just being frustrated at trying to remember all these flashcards. But and also, I just wanted to give a shout out to Ellen Cool because she was the first professor who really supported us in starting National Biomechanics Day at Stanford. And she's an incredible researcher. And I remember when I first saw her, I knew she was doing incredible work. And then when I first saw her like walking in her cow print boots. And I was like, okay, so she's brilliant and cool. I think I need to.
We called it the cool lab. Like, I mean, by definition, we were in the cool lab.
Well, could you tell us a little bit more about the current projects that are going on in your lab?
So basically, from my PhD with Ellen, the expertise that I developed is sort of, I do computational modeling. So I guess I've still not really bought into the physical messiness of biology. I do the computer stuff. But I do computational modeling of like growth and remodeling processes in soft tissues. And more specifically than that, I've published a few papers sort of mostly as co-authors on like tissues in general, like other tissues, but most of my work now in my lab is focused on the brain and the development of the brain. So big question I would say that we're trying to understand is how the brain gets its shape. So during gestation, our brains transform from this very small, smooth object. I actually usually have props that I can show now, but it's a podcast anyways, but they're also in my office. But so from this small, smooth brain around the beginning of the third trimester to around birth and of course later in adulthood, our brains are much larger and they're really intricately folded and wrinkled. And so we study the process by which that sort of wrinkled shape takes shape. So yeah, neurodevelopment, we call it gyrification or like cortical folding. So that's like the main process that we study. And we study this in really three main ways. So one, we do finite element simulations of brain growth during development. So we try to sort of recapitulate that process in our simulations. We also analyze brain shapes based on MRI images or some kind of imaging to see how the brain actually looks like, sort of quantitatively. And right now we're starting to not look only at humans but also at non-human animals, non-human primates and other animals. So to see what actually shape the brain takes, how that might change in across different species, across health and disease. And then the third area that we work on is sort of theory of instabilities, which seems sort of less relevant to what I'm talking about. But basically, the brain folding process is a biological instability. And so we also do sort of some fundamental theoretical work on instabilities that can be applied to the brain, but can also be applied to like other like engineering systems or other biological tissues that like wrinkle and fold.
Huh. That's amazing. Like it's such a diverse set of areas. And also at a scale, I think we and on topics we haven't talked about on Boom. So it's really exciting to have you as an expert in these areas. Can you talk a little bit about like why you use specific tool, like why pick FEA to do what you're doing? Like, how do you pick the tools that you're using to study these things? And
yeah, that's a good question. I recently went to a conference and saw people doing like Vertex based tools and some other things that I am not as familiar with that can also be used to track the development of biological like soft tissues. I think so. Finite element analysis, like one of the advantages is that it's just been really well developed in sort of the engineering field for a long time. The sort of disadvantage that goes alongside with that is that FEA was developed for engineering applications. So so the classic like example I always tell people if they aren't familiar with FEA is that if you've ever seen sort of maybe a picture of a car chassis and it's like crumpled or something like that, and there are like red spots and blue spots and green and things like that, like this some kind of color map, like you're probably looking at FEA results. So they're using stress concentrations or like, areas of high stress or damage or something like that. And so that's sort of the prototypical example of FEA is like, a car chassis, a bridge, some engineering device made out of engineering materials. We're talking like steel, aluminum, concrete, these very hard materials. And that's super different than the brain, which is very, very soft, right? And it's nonlinear, it's heterogeneous. So all of these things that make biological tissues complex, also exciting and challenging, those are a challenge to the applications of FEA. So within sort of the research that we do on like running simulations of brain development, there's also this whole bunch of work that goes into sort of adapting the existing tools of finite element analysis in a way that can serve to model soft tissues. So we develop our own material models that can sort of mathematically represent this addition of mass, this addition of volume that soft tissues experience, and their growth like over time, perhaps in response to like some mechanical stimulus or some morphogen concentration or something like that. So it's like the nice thing is that we have a lot of tools, a lot of literature, a lot of expertise sort of in the engineering community to draw upon. But at the same time, we have to do a lot of work to sort of cram this new application of it into this existing sort of body of knowledge.
Yeah, it's hard too, because it's hard to then validate it, right? And whereas I think about a mechanical one, or for example, we used to do FEA of car crashes, and then we did the car crash, and you can measure everything pretty exact. But with the brain, could imagine that it's quite different. It's challenging to model the different nonlinearities and the heterogeneity of it, but then it's also hard to validate that it's.
Yeah, and especially since I only do the computational work, sometimes I feel I'm like, okay, come on, guys, like get the experimental stuff up. Like I need more data, right? But it's so difficult. I mean, the brain is so soft. So I teach an introduction to biomechanics class, and in the first class, I always tell them a little bit about my research. And I'm like, okay, think of the way that you've tested mechanical properties of something before in some lab or whatever. How would you do that on the brain? If I gave you a brain right now,
and you're
and
like, actually, how would you do that?
like, how would you do that? And so, I mean, they're thinking of like an
I get
instron, right? And I'm like, okay, but how do you grab on? The brain has a stiffness very close to jello. How do you grab onto jello and pull on it, right? And your instron has like a force resolution of way more than, like, you're gonna, do one increment and it's gonna bust your like brain apart. So it's super hard. People do nano indentation on special machines that have like really good force resolution. Some people have used like 3-axis machines. Again, you need to get that, make sure you have force resolution that's relevant there. There's also some work that's been done in like magnetic resonance elastography. So doing measurements in vivo. Basically, you vibrate the brain, vibrate a head very gently, and then measure with an MRI how the waves propagate through the tissue. And looking at the speed and the phase lag of those waves propagating through the tissue, you can back out some information about the material properties. But it's super hard. So that's sort of going on the input to our models. We need this kind of data about properties, about heterogeneity, about, whatever. And then we run our simulation and we get something out. And we have to validate that, right? So, so that's one of the reasons that we got into this. Towards the end of my PhD, I got into this analysis of brain morphology, trying to understand like the sort of output of our simulations. They should be something like a brain, and how can we, make that comparison and stuff.
Yeah. I remember when I was in the lab, we were all doing scans of our own brains and looking at folding and the shape of the brain. And I can't remember exactly whether it had to do with folding or shape, but there was some hypothesis that it was related to intelligence. And I remember that Ellen Kuhl had either like the smallest brain or like the least number of folding or something like that. And she was like, well, that one can't be true. But, which that was funny. But I guess
so I didn't realize you were there for. So did you get your brain scanned?
Yeah, I did, and I built I think I ended up building a model of my own brain.
I don't think I have yours though, because I have everybody else's.
Oh. No one can have my brain.
Well, this is taking it away.
It was so tiny. I didn't want to see it.
When we got into this, we were like, okay, but we're a computational lab, but we want some data. And so at Stanford, they had a bunch of groups. I know I did mine through the economics department. They were doing fMRI scans. So I sat in a scanner for a long time and answered questions about like, would I rather have $13 now or $14.72 in 7 weeks and things like that. Well, they like watched the activation like light up in my brain. And yeah, so I did it and they paid me. And then I also said, can I get the data? And so I got the scan of my brain and a bunch of, I think I have 12 brains of sort of like, Ellen, me, lab mates, things like that. And it was just sort of a way to like bootstrap our first analysis, like while I figured out like what to do with this and stuff. And yeah, so I wonder if you were actually at this lab meeting where I think I presented some results. And if I remember correctly, it was Michael Ortiz who was visiting. And he's a professor at, I believe, Caltech. He was visiting, and I showed all this data, like the brain volume and the surface area and the gyrification index, which is sort of a measure of how folded the brain is, and blah blah blah. And yeah, I actually get this question a lot. So I would say that intelligence is sort of related to foldedness on like a species scale. If I gave you a bunch of animals and asked you to line them up from like smartest to dumbest or whatever, like intuition would map sort of roughly onto like foldedness. Okay, so more intelligent animals have higher folded brains. So like us, dolphins like have really highly folded brains. Like, yeah, rabbits don't. Okay, they have almost smooth brains. So roughly on a species scale, yes. But anyway, so I knew this. There's like a lot of other things going on there too. So regarding brain size, I think maybe Ellen's was the smallest, I don't remember, but it's also brain size is proportional to body size. So women tend to have smaller brains. And then there's also aging effects. So like Ellen was older than like all of the students, right, that did it. So at this lab meeting, I presented this data, just showed a bunch of data points. I was kind of interested in like, okay, if we say, the gyrification index of a human brain is 3, does that mean that everybody is like 2.98 to 3.03? Or is it like, we're talking like 2.5 to 3.5, right? I wanted to get an idea of like what the spread was like.
Yeah.
And so Michael spoke up and he said, I don't think that you're aware of the history behind this. And if you were, you wouldn't be joking about it in this way. And I was really taken aback. And he suggested that I read a book called The Mismeasure of Man by Stephen Jay Gould. And I did. I'm not sure if either of you are familiar with it. And I really understood like where he was coming from. And I think it was an important thing for me to be aware of, like working in this field. Basically, Stephen Jay Gould was writing sort of a response to the bell curve, which was this other book that was making assertions basically about, it's basically a foundation of like scientific racism. So these
like
races
what is normal?
Have smaller or less folded brains. Women are not as intelligent as men because they consistently have smaller brains. So it's clear that they can't be, trusted to vote, right? Or, things like that.
Yeah.
So this has a long, long history, right? This scientific racism, specifically in the study of the brain. So it was an awkward moment for me to get called out on that, but I think it was really formational for me. And actually a couple of years ago, I bought this book for all of my grad students. So that we could sort of discuss about it, because I think I do get this question a lot. So like, was Einstein's brain more folded and things like that. And so what I would say is that on a species level, a really gross level, when you're talking about huge changes in folding, there is sort of some parallel with foldedness and intelligence, but on like an individual within species level, I mean, a real challenge is we also just don't know how to measure intelligence, which is hugely fraught, and like a third of that book. So anyways, that's a little like lesson that I had to learn in like I said, a little bit of an uncomfortable way, but I really appreciated him sort of pointing that out to me, that blind spot.
Yeah, and I think that's a great reminder. I feel like it is hard when we get like, I mean, I've done that, so you kind of like feel like your foot's in your mouth, but I think it's important, right, to think about these things sensitively, and also like a reminder about like, science is so human. What are the ethics of what we're doing? What are the implications? How does this affect our identities? Like all of those important questions as we're getting excited about the data that we're seing, right? Also like thinking forward. So yeah, thank you for sharing that. And thanks for sharing. That's awesome that you share that book with your lab. That's really cool. So I guess so switching maybe to a parameter that you guys have found is really important and significant in your biomechanical analyses, and was the big subject in your NSF Career Award, talking about cortical thickness and sort of how that relates to to important functions and things like that. Yeah, can you just tell us what's known about, well, what is it? What's known about it, and why do we care about it?
Yeah, so the cortex is the thin outer layer on the surface of the brain. If you look at an MRI, it's like sort of the darker gray. It's called gray matter also, as opposed to the inner white matter. And I often make the analogy that it's sort of the computational power, the processing part of the brain. And so the interior part, the white matter, is like full of cables that connect the different sort of CPUs in our brain. So cortical thickness, the cortex is important in general. We have this folded brain because it allows us to pack more cortex, more surface area sort of, into the constrained volume of our heads. That's why we have folded brains.
Biology, right? All about optimizing surface area volume.
Right. It's a huge pattern that you see like across biology, right? So cortical thickness, we're looking at the thickness of this layer. So it has this hugely expanded surface area throughout the brain. Cortical thickness, I think, is just very interesting. Like there have been these like allometric studies looking at species across huge ranges of body size. I want to say it was 6 orders of magnitude in body size that we found in a paper. We didn't find this. And so like surface area and volume change also by like 6 orders of magnitude, and cortical thickness changes by like 1. And so there's clearly some kind of control going on there. Like this is an optimal thickness that we've sort of settled on. So I find it interesting that the cortex doesn't vary in thickness much between, let's say, a shrew and an elephant, but it actually varies quite a lot within an individual. So this part of your brain versus that part of your brain versus this part of your brain actually varies like between, I would say, like 2.5 to 3.5 would be like even a standard deviation, like a millimeter. But I mean, sorry, yeah, this is in millimeters, but that's like
significant.
But that's like almost 50% variation. So it's pretty significant. And so variations in cortical thickness are associated with a number of different neurological disorders, like epilepsy, schizophrenia, autism spectrum disorder. Often when people talk about cortical thickness, they're talking about like a single number for like your brain. So it's the average of your cortical thickness, like across your whole brain. But like I said, it actually varies really significantly within a single brain. And it actually varies in a systemic way. So in these folds, the valleys tend to be much thinner than the peaks. So they're called sulci, or the valleys, and the peaks are called gyri. And so gyri are consistently thicker than sulci. And so we showed in a paper towards the end of my PhD that this thickness difference is at least partially caused by the folding forces, like just the act of folding the cortex sort of makes the sulci thinner and the gyri thicker. And so in my career award, we're trying to understand better the role of forces in these thickness differences, and specifically look at the role of mechanics in these changes in thickness across different species and throughout this really important period of human development, like during gestation and the first couple of years of life.
That's so interesting. And I'm curious, when in your career award too, I think you mentioned that you want to study modified cortical thickness, so removing this effect of folding on analyzing and comparing cortical thickness. We're wondering how you came up with this method and what sort of insights do you hope to gain from taking this approach?
Yeah, so the idea behind modified cortical thickness is that if we have, we know that there are 2 things that are contributing to the cortical thickness. There's the mechanics of folding, but then there's also like everything biological, right? Cells doing their thing, genes doing their thing, cells doing their little ECM thing, right? Like there's a lot going on there biologically, which we believe is probably more important functionally to how the brain works. Like are, for instance, neurons making a lot of a large dendritic arbor, so a lot of connections to other cells that would affect the function, and would also like affect presumably like the size of the cortex in that area. So we have these sort of two factors, and if sort of biologically or clinically we'd be interested in one of them, our goal is to sort of calculate the effect of the mechanics and remove that so we can see only the result of sort of the biological stuff. So we're still working on the details. That's aim one of the proposal. But yeah, that's the idea, is if we can sort of remove that effect of folding. And the reason why that's important, and the reason why we're looking at these two cases of sort of evolution and development, is that evolutionarily, if you want to compare, for instance, a human and a chimpanzee brain, so it's sort of a big question is like, what makes us human? Like, why are we having this conversation right now and not like or
dolphins?
Right? Yeah. And so if you want to compare these brains and, for instance, find like the seat of like vocalization and like what, makes these gives us these abilities or something, you're trying to compare like, this region of the brain in a human and a chimpanzee. But those regions are differently folded in a human and a chimpanzee. So if you want to sort of compare them functionally across different folding, then you're going to have this confounding factor of the mechanics of folding. So we want to be able to take that out. And similarly, if you're looking at sort of along development and you want to track the development of cortical thickness in an area, well, it's both, developing in the biological sense, but also folding. And so you're going to have, again, that confounding effect of folding, sort of changing your results along the time. And so if we can sort of remove that effect, then we can compare more apples to apples at different times or across different species. So that's the main idea of my career award.
Wow. And how do you remove that effect?
So we're trying to use a machine learning approach. So we have simulations where we know, that's the nice thing about simulations is you sort of know everything involved. You know the inputs and you know the outputs and any kind of output that you want. So we're running simulations where we know sort of what is happening like biologically, like volume change, mechanically induced volume change and growth, or like sort of morphogen induced. And we can see both the actual thickness that comes out of this and the modified cortical thickness. We can calculate that as well. And so we can train a neural net on these simulations and then apply that to some data from MRIs or some kind of imaging data.
Oh, cool. Yeah, it's amazing. Like, I feel like Melissa and I have both done a lot of experimental work, and like we were talking about the messiness of an experiment, and like sort of the beauty of a simulation and advantages of a simulation is that you can do things that you might not be able to do in an experiment.
Right. You can just probe from like all different levels at any different time. Like, you never have this sort of trade-off of like, okay, we have to sacrifice, an animal to like get this time point, so there's definitely huge advantages there.
Right. And cool that you can look at things like evolution, which we can't, we can study using a fossil record or, whatever exists now, but like you can really simulate that timeline. Or how that might be impacting. Well, we want to like also make sure that we have time to sort of switch gears and talk about your outreach, because I think that really spoke to us, and we really appreciate people that not only are doing amazing research, but are also really passionate about translating that to broader communities, getting other people involved, and that just really shines in your career and in your work. So can you just tell us a little bit about that? Like, I know it was a big part of your application, your career application, as Melissa mentioned earlier, like you mentioned Boom in it, so obviously we want to talk about it. But what is outreach meant for you, and like how do you make that part of your lab and your life?
So I think like just early on when I got into biomechanics and started studying the brain, I think it was honestly a little bit frustrating for me that a lot of people have this idea of biomechanics as being sort of like whole body motion, like muscles and bones, like, and like that is biomechanics. And I, opened this door into this world where like everything is biomechanics, right? Like, I get so much surprise when I tell people I'm an engineer studying the brain, and not that kind of engineer, like I'm a mechanical engineer. But like, so I've like been on papers on, the brain, on skin, heart valves. And so like, every part of our bodies is interacting with and experiencing forces, like, and that's what biomechanics is, like, period. So I just got, like, sorry,
I just said period.
period. And like, so I got this like glimpse at this like wide world of biomechanics, but I feel like people don't know about it. And so when I'm just like walking around in my normal life, I mean, especially now as a mom, like, oh my gosh, the biomechanics of children and babies is, well, and childbirth and pregnancy, like all the way through, has been a wild ride. And there's like so much going on there. But people like most people don't think of it as biomechanics. And so when I started to teach this introduction to biomechanics class, I wanted it to be like a buffet. Like, I'm going to put out everything that biomechanics is, and you're going to get a little sampling of it, and you can go back for more in like whatever area that you really liked. And so I started this project that I called Biomechanics in the Wild. And basically I told students, like, I want to prime you to see biomechanics around you. So I want you to like think about the things that you're interested in and think about like what connection is there to biomechanics. So like some sport that you do, or some disease that a family member has been struggling with, or like some phenomenon, like why is my best friend's hair curly and mine is straight or something, right? Like there's just so many interesting questions out there. So it took a couple iterations, but we ended up the status of the project as it is now, and this was basically the education and outreach part of my career proposal to sort of continue developing this. So students pick a topic that they're interested in, and they find some original research papers that are related to that topic to learn about it, and then they have to share that information. So they share it in multiple different formats. So they write a technical report for like an expert audience like me, because I think that is, of course, an important part of science. But it's also important that you can talk with people who do not have a PhD in engineering, right? And so we also have a part where they do, originally it was just a blog post. Now they can do a podcast episode, sort of inspired by you guys, or a YouTube video to share this topic with like the general public. So I tell them, nobody who has like a STEM degree, right? And so it's just been really cool. I think they get really into it. They really seem to enjoy this like science communication part, definitely much more than the technical report. And they've done a really good job with it too. And so in the career proposal, I said, basically, I want to sort of continue developing this to give students better training and like engaging with the literature and then communicating that to different audiences. But then a second benefit is that the public can learn more about biomechanics. So we have, it's I think sites.nd.edu/biomechanics in the wild. And we have like, I think over 100 short articles. They're like 500 words that are just really cool stories about carnivorous plants and bat flight and how archerfish hunt and figure skating and like ACL repair. And I mean, just anything you can imagine. So we have that. And then the third part of this outreach proposal is that there's not actually anything that's specific about this idea to biomechanics. I think that probably a lot of fields have similar challenges. Like as an expert, you see your topic everywhere and other people don't see that. So I think that it could, I'd love to expand this to sort of something like STEM in the wild, like with different disciplines, like I think statistics or immunology or nutrition or linear algebra, right? Like the stuff is all around us. And so to help with this sort of twofold aspect of educating students and then educating the public. So yeah. So I had reached out to you guys. You actually wrote a letter of support. Thank you. I'm sure that's why the grant was funded. Um, and so I just, I think that Boom is this other, like, this really great public facing initiative to like increase awareness of biomechanics. And I just thought there should be some synergy there. I don't hear you guys right now. Yeah, I saw you laugh, but I didn't hear you laugh earlier. No, I'm not hearing you.
Well, we have been really inspired by your outreach, and I've loved reading the Biomechanics in the Wild articles, and it's so cool to see how you're involving students in that too. And we'd love to think of more ways to collaborate in the future with these projects. So
Yeah, for sure.
that would be really exciting for us. Could you tell us about shifting gears a little bit?
Yeah, so on that note.
Okay, usually we have better transitions.
That was perfect.
Thank you. Yeah. So one of our favorite questions is talking about failure. You talked about also not receiving the NSF Career Award the first time, but going for it again and receiving it the next time. So that is really, I think, always something nice to share that isn't always shared, but I think makes us feel like
feel better.
Yeah. Feel more inspired for us to overcome obstacles or be more.
Oh yeah, I mean grant funding is just ripe with failures.
Yeah, well, would you be willing to share? I don't know if there's any other failures that come to mind, but we'd love to learn about sort of an experience of you feeling like you might have failed and what you learned from it.
Yeah, I was preparing for this question, and actually I came up with a really great example. So I had a new student start in my lab in 2020. And I mean, just kind of a mess of a year, right? And on top of that, he started like literally, I think 3 or 4 days before I was induced with my son. And so I needed to come up with a project for him, and I was just like, okay, I don't know, look into this, right? And when I came back from maternity leave a few months later, like he was showing me the data, and it occurred to me like the question I'd asked him made no sense. I was basically asking him to like measure something that we were controlling as an input. And I cannot express to you how stupid I felt. I mean, I felt like I was very disappointed in myself, like for, I felt like I had sort of wasted his time, which was like weighed very heavily on me. And also just like kind of embarrassed. It seemed like such a rookie mistake. So I think, failure is an unfortunate side effect or happenstance like along the road to often better things. So the good news that came out of this is that so he developed in sort of analyzing the simulations that I'd asked him to analyze, he developed some really nice code that we've used since in some other students' projects that are not as meaningless. So the time was not completely wasted. I mean, and of course, like he learned things along the way, right? Like
of course, yeah.
Learned about the project and about the question and about the code and everything like that. But we have been able to use like his actual output from that. And then the other thing is it just got him like really interested in the project and like really interested in these questions. So he ended up becoming the main contributor on this perspective paper that we're hoping to submit actually really this month, which I'm super excited about. So this there's this like foundational paper in sort of the field of brain morphology from 1929, sorry, by actually he's a Dutch neuroanatomist, but written in German. And everybody cites this paper, but like most of us don't speak German. I mean, Ellen Kuhl actually does, right? But so there are some people who probably read it and enjoyed it in its entirety, but a lot of us haven't. And so I hired a Notre Dame student. I found a chemical engineering German double major and paid him to translate this paper. And so we're going to be submitting the full English translation of this paper that a lot of the field has been built on, but I think has been often built on sort of our understanding of the figures, not really the text. And then we wrote an accompanying perspective to sort of explore what were the ideas at the time that he was responding to, and how has that gone on to influence the field. So he got really just into the question and really put a lot of work into the perspective paper. So I think it turned out well, but I felt like such an idiot. I cannot even describe.
It's hard when it's someone else's time. I feel like that's also where my main failures feel it's oh no, it's not just me. I don't care if I fail sometimes, but if it affects someone else, it's hard.
Yeah.
So totally can
Yeah, and I can look for the bright side, oh well, I learned something from it,
Yeah.
and but when I have to tell him that hey, you learned something from my mistake.
Yeah. Together. Yeah. No, but thank you for sharing that though. I think, and that also normalizes even professors make mistakes. Sometimes I think as a student, you're looking to your professor for all the answers. And and sometimes I felt it really, really refreshed when your professor just says, I don't know. And you're like, okay, thank God, because I don't know. Yeah. So thank you for sharing that. So as we wrap up, I'll just ask, how can people follow you in your work? You mentioned how they can follow biomechanics in the wild, but yeah, what your social media handles, whatever you can share.
Yeah, so I think the best way is my lab website. I have actually a publication page I'm really proud of. I have word clouds for all of our papers. They look pretty cool. And also, so on Twitter, I'm not super active, but I'm there. Holla back, Holla_back.
That is bananas.
I've gotten some compliments on the Twitter handle.
Yes, Hannah. That's awesome.
Yeah. So and I'd be happy to hear from people.
Okay, great.
Great.
We'll share those in the link in the description of the episode. You just have to take a pause to commemorate
Appreciate
Glenn.
that. Yeah. Well, we want to end with our favorite question to end.
Oh no, I lost you again. I mean, I think I know the question if you want me to answer the question. And you can add your voices in later if you want.
Sorry. Third time's the charm.
Here we go.
All right. Well, we're now it's time for our favorite send-off question, which is, what are you most excited about for the future of biomechanics?
Yeah, I think I'm most excited just for all of these. I mean, I'm going to put new and unconventional in quotation marks, but these not sort of the areas that are not sort of these traditional staples of biomechanics. I mean, I'm of course really interested in soft tissues, but there's so many soft tissues. So just some of the tissues and organs that people don't think about as being mechanically important or something. So two examples of this that I think really capture the idea. So mechanics of the lung, here in year 3 of a respiratory illness pandemic, the lungs are something that are super important to us, very important mechanically and hugely understudied. So I have a colleague, Mona Eskandari at UC Riverside, who studies that, and I think it's just so important. And another topic that I think is really interesting and just really exciting, and people have been working in this area for a little while, but it's sort of, I think, taking off more and more every day is reproductive biomechanics. So there's so much going on in like the reproductive organs, during like all the time, but especially during pregnancy and delivery and recovery. Those tissues, are under the most ridiculous mechanical stimulus and constraints like imaginable. And it's fascinating, hugely important to every single person who's been born. And so there's a lot of good people working on that stuff, but I'm just really interested to see that field develop as it gets maybe more incorporated into like mainstream medicine and clinical work and stuff. I think it's just really important work that needs to be done.
Yeah, talk about like us being in awe of biology, like, excited rather like the messiness of biology. I feel like development.
And talk about messiness.
Process, you're like, wow, like how?
Yeah.
But it's a carefully orchestrated mess somehow.
Yeah.
Well, thank you for sharing. And like, I think those are both new answers to that question that we've never had before. So
well, you should check them out and have some of those people on maybe.
Yeah, we will. Actually, yeah, that would be great to have those people on. And just thank you for sharing your experiences and your stories and your journey. It's been such a fun ride and just so great to have you on the podcast.
Yeah, thanks so much for having me, and let's definitely keep in touch on this sort of goal of like outreach about biomechanics. But I think it's so awesome to see the cool stories that you guys have shared, and the longevity and success I think that you've had is just really, I find, very impressive. So
thank you. Well, yeah, it's definitely due to all of our support and then having amazing guests on like you. So yeah, thank you.
Biomechanics off our minds.